4,000 meters down, the ocean stops pretending to be survivable. No light, no warmth. Pressure so absolute it would crush a submarine hull into scrap metal in seconds. The cold alone would stop a human heart before the first minute ended. Nothing should function here, and yet, something does. In this void, where the physics of survival seem to break down completely, biology has written a different set of rules. Cells that should fail don't. Tissues that should degrade persist. Time itself seems to move differently for the creatures adapted to this darkness. And among them, one animal has taken this to an extreme we're only beginning to understand. The Black Devil Anglerfish. A predator no larger than a fist, drifting through absolute blackness with a lure of living light. But it's not the hunting that fascinates researchers. It's how this animal exists at all. At these depths, proteins should misfold. Cell membranes should destabilize. The machinery of life should slow, then fail. Under pressure that would flatten steel. And yet, the anglerfish persists. Its cells remain stable. Its tissues hold together over long spans of time, in conditions that destroy most biological systems within hours. Something in its physiology has adapted not to fight damage quickly, but to prevent breakdown in the first place. This isn't a cure for anything. It's not a blueprint humans can copy. It's a system that only works under conditions we cannot survive. But it raises a question worth asking. What happens to biology when you remove every constraint we thought was permanent? At 4 kilometers down, cells forget how to age. Researchers call the emerging hypothesis pressure-locked repair. The idea that certain deep-sea organisms maintain cellular integrity not despite extreme pressure, but because of it. The Black Devil Anglerfish may be the clearest example we have found. Its cells show persistent error correction. Its telomeres do not shorten on schedule. The biological countdown that defines aging in surface organisms appears to operate on a different timeline, or barely operate at all. Imagine a clock submerged under a column of water taller than 10 Empire State Buildings stacked end to end. At that depth, even time bends. This is not magic. This is evolution, solving a problem under constraints so extreme that the solution cannot exist anywhere else. If you know Dune, you already understand the shape of this discovery. Pressure is the desert. The anglerfish is the sandworm. What lives inside it is the spice, powerful, transformative, and locked to an environment humanity cannot inhabit. Here's the observation that started everything. Deep-sea tissue samples from Melanocetus johnsonii showed cellular structures that were not degrading normally. The telomeres, the protective caps on chromosomes that shorten with every cell division, were being actively maintained. This should not happen. In every surface organism we have studied, telomere shortening is inevitable. It is the clock. It is how cells count down towards senescence and death. But in the Black Devil, something was interfering with that countdown. The cells were not immortal. Nothing is immortal, but they were stable in ways that surface biology cannot replicate. The mechanism is not the fish itself. It is what lives inside the fish. The Black Devil Anglerfish hosts a bacterial ecosystem, dozens of species, possibly hundreds, concentrated in its liver, its gonads, and its bioluminescent lure. These bacteria exist in symbiotic relationships so tightly co-evolved that separating them from the host kills both. Together, they produce an enzymatic cascade. Not a single compound, a system. Multiple molecular pathways working in concert to stabilize cellular repair that would otherwise degrade over time. The fish provides the environment. The bacteria provide the maintenance. Neither survives without the other. Here is the constraint that makes this discovery both profound and inaccessible. The enzymatic cascade requires pressure. At 3,000 meters depth, the water column exerts roughly 300 atmospheres of force on every square centimeter of tissue. This pressure does not just fail to destroy the bacterial proteins, it appears necessary for them to fold correctly. At surface pressure, the proteins denature. The bacteria die within hours of decompression. The system collapses the moment you try to remove it from its environment. Pressure-locked repair is not a resource that can be extracted. It is a process that only exists under conditions we cannot survive. Evolution does not have goals, but it solves problems. For 400 million years, in permanent darkness and crushing pressure, this symbiotic system developed. It optimized for an environment with no solar radiation, no oxidative stress, no temperature fluctuation, stability at the molecular level. The result is a biological proof of concept, evidence that persistent cellular repair is possible under the right constraints. This matters for research not because we can harvest the bacteria, we cannot, but because we can study what they do. We can identify which mechanisms might be adapted or approximated for surface biology. The deep ocean is not offering us a cure. It is offering us a question we did not know how to ask, and science is finally developing the tools to pursue it. Every attempt to culture these bacteria outside the anglerfish has failed. Every attempt to maintain anglerfish in pressurized laboratories has failed. But failure is data. Each attempt teaches us more about what the system requires. The next generation of deep-sea research, long-term observation at depth, pressurized habitats, in-situ study, is already being planned. This is slow science, expensive science, science that resists shortcuts. It is also science worth doing. The same pressure that enables the phenomenon is what makes it untouchable. The same constraints that allow the bacteria to stabilize repair are what kill them when we try to extract them. The same depth that created this system is what keeps it beyond casual reach. Pressure-locked repair exists because the environment demands it, and it will only continue to exist as long as that environment remains intact. We have found something extraordinary. We cannot take it, but we can learn from it, carefully, patiently, on its terms rather than ours. The Black Devil Anglerfish will continue to drift in darkness, carrying passengers that rewrite the rules of cellular time. The pressure will hold, the bacteria will persist, and we will keep looking, knowing that the ocean solved the problem we are only beginning to understand. The door is open. The work has just begun.